This method provides a complementary preclinical approach for studying the immediate cellular response to blood in the brain following a hemorrhagic stroke, a very serious condition for which we have no specific medications available for patients.
Unlike rodent models, the transparency of zebrafish larvae allow us to observe cellular responses within the brains of live, intact animals in real time using fluorescent microscopy.
To begin, use a tea strainer to collect all fertilized embryos from natural spawning in breeding boxes produced from 1 male and 1 to 2 female adult zebrafish. Transfer 100 embryos to each petri dish containing standard E3 embryo medium. Incubate at 28 degrees Celsius and stage according to standard guidelines. At 6 hours post-fertilization, remove dead and unfertilized embryos from the dish using a Pasteur pipette and put the dishes back into the incubator. At 24 hours post-fertilization, under a bright field stereo microscope, use sharp ultra-thin dissection forceps to decorionate embryos for atorvastatin treatment. Then, add 30 milliliters of E3 embryo medium to 2 clean petri dishes, one for treatment and the other for control. Remove 60 microliters of embryo water from the treatment dish and add 60 microliters of 0.5 millimolar atorvastatin to achieve 80% of larvae hemorrhaged. Using a Pasteur pipette, transfer 100 embryos in as little water as possible to each dish. Incubate the 2 dishes at 28 degrees Celsius. At any time